use a lot for the diamino-benzidine immunohistochemistry. And a new thing that we've been starting the last actually several years now is a technique to label neurons. So I'm a neurobiologist. We work with brain tissue pretty exclusively. And so there's a technique to label cells using a very old technique developed back over 100 years ago called the Golgi-Cox technique, which again I'll describe momentarily. We use immunohistochemistry at the EM level. We use diaminobenzidine as a chromophore. We also use another chromophore called VIP, which I'll show you what that looks like. And then a lot of the work deals with post-embedding immunogold for either glutamate or gamma-aminobutyric acid. Again, I'll show you some examples of those. One of the reasons why we really love the BioWave is the fact that if we want to cut fresh brain tissue for Western protein immunoblot analysis, we could use the tissue from the brain and then we can BioWave the rest of it to do immunohistochemistry. Primarily, we do that at the light microscopic level, but as we have found, the tissue preservation is surprisingly good even at the EM level. Even after we cut the tissue, we keep it in the cold, then we process it in the BioWave for 90 minutes with fixative, with EM fixative. And then when we process it for EM, the tissue preservation is amazingly good. So although I typically perfuse the animals with fixative, you can actually get away with doing EM of freshly dissected tissue and then process it for EM. It's really quite amazing. Again, we can use fresh tissue, dissect the areas of interest, use the BioWave for 90 minutes, and again, we'll show you some of the examples that we've done in the past. Because of the fact that we want to get good ultrastructural preservation, we've had to balance between using formaldehyde or paraformaldehyde versus glutaraldehyde. And because of the fact that, because we do a lot of post-embed immunogold labeling, we need the glutaraldehyde. So we've had to kind of suck it up in terms of the balance between getting good light microscopic immunohistochemistry and the post-embed immunogold. And again, because of the BioWave, we're able to get away with this because it takes so little time to actually process the tissue, as you will see in a moment. Again, this is the BioWave that we use. The steady temp is on the opposite side over here. We perfuse the animals with a glutaraldehyde, paraformaldehyde, picric acid combination. Then, if it's fresh tissue, we'll BioWave the tissue for 90 minutes. We'll post-fix the next day in 4% paraformaldehyde. But the key thing here that we have found is really using that steady temp. Because in the past, what people have done is they would just put the tissue in vials and then nuke the vials. Okay, well, we started doing that, of course, until we could afford to buy a steady temp. But once we started with a steady temp, what we do is that we have this tray that, again, we buy from the company. We have these holders that we put the brain tissue in, and because the fixative is flowing through here at a steady temperature for, in this particular case, where we do 30 minutes after we perfuse the animals, we take out the brain, and then we post-fix for 30 minutes in the BioWave with the steady temp, with the fixative flowing through this box here, in which the fixative is flowing through, again, at a steady temperature. So that's really the key that we found that makes the tissue preserved incredibly well in our hands. And until then, we were getting variable fixation, preservation of the tissue if you just nuke the tissue in a vial with fixative in it. The steady temp has been an amazing advance in terms of having the fixative be able to flow through here, this contraption here, and being able to and being able to, oops, sorry about that. Being able to flow through this box here at a steady temperature. I mean, that's been really one of the keys in terms of getting really good ultrastructural preservation.
Isn't the steady temp supposed to flow through the cold spot? No. No?
Cold spot.
You can do that.
It can for different purposes,
Oh, okay.
but for their purposes, no.
Okay.
Okay, thank you. I mean, we just use it for this, and in fact, we do, because I'm a neurobiologist, we do a lot of brain, a lot of animal perfusions, and we use this like all the time. We use this like all the time. So the key thing that Cindy really came up with was after we slice the tissue with a vibratome, we go anywhere from, again, 40 to 60 microns. The question is, how are you going to be able to process the tissue? Well, it turns out that you cannot use well plates because the well plates are heated up and you'll fry the tissue. Okay, so we found that out early on, and Cindy, through her being able to get onto the internet and find these little dishes, we can process up to 40 brain slices at a time. Plus, of course, as you can see here, we can put again, this is mouse. So we can put multiple brain slices in a given well, in a given dish, okay? Again, on the website, it has the name of the company. They're amazingly inexpensive. They work amazingly well. And again, in terms of volume, which is something I didn't include here, we could use as little as...
For the smallest ones, 100 microliters.
So we can use, in the smallest plates that we have, you can use as little as 100 microliters of antibody to process the tissue.
The bigger ones are about 500.
Right. The bigger ones, in which you can put, 6 to 8 pieces of tissue at one time. With these smaller ones, we put far less. And again, you can get away with 100 microns, 100 microliters of volume, which is really astounding. And so there's a company that sells these. They're relatively inexpensive. And so we go through this protocol, which again is on our website. I don't have to go really through the details, but we do this for both electron and light microscopy. And these little dishes have just been the most wonderful thing in the world for us. Again, this protocol is on our website. Without going through a lot of detail, one of the key things when doing immunohistochemistry, both at the light and EM level, is this antigen retrieval step. In the old days, you would end up basically boiling the tissue. Okay, well, you don't have to do that in the system. The antigen retrieval step, we either use pH 6 or pH 9, depending on what will be best for the antibody. We found that you cannot say that each antibody will work well at pH 6 or pH 9. Whenever we get a new antibody, Cindy tests it at both pH 6 and pH 9, a couple of different dilutions, and figures out which one looks the best. So this antigen retrieval step, again, takes 5 minutes. You go through all of the usual rinses, primary antibody, secondary antibody. Again, the primary antibody, we do 36 minutes. Secondary antibody is for 16 minutes. If you're doing immunohistochemistry with diaminobenzidine, there is the ABC there's the ABC step, in which, again, one of the other advances that we found, again, when Rick Giverson was involved in helping us with this, is this compound called imidazole. This has been an amazing advance for us. And the funny thing is that in the book that Rick was involved in writing a chapter for, it gave us imidazole, but it didn't give us the recipe. So luckily we found out the name of the chemist at Chico State. We called him up and he was at his desk at the time. He went into his notebook and gave us the recipe for the solution. I mean, it was really amazing. So this whole process takes 85 minutes instead of having to incubate overnight. So this whole process takes 85 minutes. It's truly amazing. Processing for EM, again, we don't have to go through the details. If you do any sort of EM, it's the usual stuff with osmium and urinal acetate, ethanol, propylene oxide, resin. This whole process takes 41 minutes. What we do is we do cure the samples in the oven overnight. We've kind of tried using the BioWave a little bit for this. We haven't done a lot with it, and because we do a lot of flat embedding of the tissue, we will flat embed it like this. And let me just show you. This is Aclar.
Yeah.
This is Aclar, and we just simply flat embed it, and then we can cut out a piece of tissue, embed it on glue it on a block, and then thin section. So again, it takes relatively little resin, as you can imagine. So this whole process takes a whopping 41 minutes to do.
Do you fix first
Yes.
overnight, or do you fix in the microwave as well? Cindy, do you?
In the microwave.
Okay.
In the BioWave.
Okay.
Yes, that one I do.
Okay.
Everything else I don't know. But,
Okay.
no, that one. I mean, again, we used to fix overnight when we weren't doing any pre-embed immunohistochemistry.
Yeah.
But with post-embed, we fix overnight. Everything worked well, but with pre-embed, you can't do that. So we've had to compromise between getting good pre-embed immunohistochemistry and getting good post-embed immunogold labeling, because the two of them are diabolically opposite from each other. So we've kind of bitten the bullet on both and have decided to compromise on both ends. So the post-embed gold isn't quite as good as what we used to have. The pre-embed isn't quite as good as we've gotten away with in the past without, for example, either using no glutaraldehyde, but the ultrastructural preservation is always like the most amazing thing. So, so that's with the EM embedding process. Again, we can do double labeling for different chromophores, for DAB and VIP, and I'll show you an example of that in a moment. Golgi-Cox staining, again, we get relatively good labeling with this protocol. Again, we'll show you a little bit more in a moment. And then we've just started doing some more fluorescent labeling, again, with the BioWave in terms of the tissue processing. We can do double labeling, which again takes about double the amount of time. Essentially, it's really exactly the same procedure as the single, but you just do it second time with a different secondary antibody, essentially. But again, only about 3.5 hours. It's really an amazing accomplishment. So this is the double labeling that we've done, where we've done both DAB labeling, which is the darkened product here in kind of the background. The VIP labeling are these globs, these rosettes here. So in the next slide, Cindy has outlined what the VIP looks like. And then again, without it, you can see this is the VIP labeling here. This is the DAB labeling in the background. So you can actually do double labeling, pre-embed double labeling. The Golgi protocol, again, this was based out of a procedure that came out in 2013. The total time is a little bit over, is overnight plus an extra hour. Again, without going into a lot of detail, this is, again, a protocol that has been around for over 100 years. But the point is that we've been able to modify it. We, meaning Cindy, has been able to modify it for just overnight. And again, in a couple of slides, I'll show you the comparison between the two. The Golgi, again, here's a typical Golgi-Cox procedure where you can see these little spinules, which are spines. These are nerve fibers. These are nerve dendrites, and you can see the spines coming off of that very nicely. With fluorescence, again, we've just started doing fluorescence microscopy using the BioWave. Again, a little over 2 hours. Essentially, the procedure is pretty much the same. Just takes a little bit more time.
That's because that's for two different
For two different antibodies. Ah, got it. Secondary. Okay. All right. So if you're only doing with one antibody, cut this in half. So it's about the same. It's about the same time. Again, the antigen retrieval step is really critical in the sense of determining what pH to use. Really critical. pH 6, pH 9. Each antibody is very, very different, so you have to really go through a test protocol first. Our fluorescence that we're starting to use in the lab, again, comes out very, very nicely using the BioWave. And especially with these new transgenic animals that are constantly coming out, and the animals are, of course, very expensive. We've had to really make sure that we can utilize as much tissue as possible using the BioWave. Again, just a few examples from collaborators of mine, Chuck Allen at OHSU and also Vivek Uni at OHSU. The specifics are not really that important except for the fact that when we do pre-embed immunohistochemistry, looking at GFP labeling, for example, at the EM level, you can see very nice diaminobenzidine immunohistochemistry. We do post-embed gold for glutamate, so that's what the little gold particles are here. And again, just another example of that here. Again, tissue preservation is really quite good. And again, you're really saving a tremendous amount of time by using the BioWave. One of the interesting things, now GFP is fairly easy to work with, admittedly. It's a very strong antigen, very good antibodies available for it. Receptors are always a real problem because they're membrane bound. They're much more sensitive to the fixation. But again, because of the BioWave and the fact that you're really not exposing the tissue all that long to the fixative, the immunolabeling comes out amazingly well. This happens to be just a particular receptor that my colleague Chuck Allen was interested in. And again, at the EM level, you can see nerve terminals labeled here. You can see receptors within the cytoplasm of the cell body labeled. And you can see a flow up here. You can see the labeling here. And again, this is post-embed labeled for GABA. So again, you can get both pre-embed immunohistochemistry to work with the receptors, post-embed immunogold to work for GABA. I mean, it's really quite amazing. And again, the key thing is that you're really exposing the tissue for a relatively little amount of time compared to the traditional methods. Again, just another example with collaborator Vivek Uni. Again, GFP labeling. Again, GFP is just very forgiving of a lot of stuff, to be honest with you. This was a protein called alpha-synuclein. Again, it's not important what the name of it is. That was GFP labeled. Again, using pre-embed GFP labeling, we can see DAB labeling here in the terminal, post-embed gold for glutamate, synapsing on a dendrite here. So you can see the specificity of the GFP labeling right here. Again, ultrastructural preservation is really quite marvelous, considering. I think probably one of the most important slides here is just a comparison of the different methods. Microwave or BioWave used versus bench. For typical EM tissue processing, the BioWave is 41 minutes. Again, we put it in the oven overnight so that we don't have to hang around. The key thing here is that on the bench it takes at least about 22 hours. So incredible difference between these two. Pre-embed immunolabeling with the BioWave, it takes 45 minutes. I'm sorry, 85 minutes. Sorry about that. 85 minutes compared to a good day and a half with the traditional benchtop, where you're incubating things overnight, either in the primary or even in the secondary. The Golgi-Cox technique, this is probably the most stunning thing. So when we first were planning on doing it, we actually did this procedure for a couple of weeks. It takes anywhere from a week to 2 weeks, maybe a little bit more. Of course, Cindy said there's no way she's going to wait for 2 weeks or 3 weeks for this to happen. So she says, okay, let me start looking at the BioWave and see if it will work. Well, we've got it down. Well, the original technique was about 5 hours. It's now a day plus. So again, we're talking about 1 day really versus, we need to update that part. 1 day versus anywhere from really 1 week to 2 weeks. So if you're really interested in that kind of morphology, obviously the BioWave is going to work significantly better. And again, it's really the exposure. You expose the tissue for relatively little time during the entire protocol. It's 5 minutes here, it's 30 seconds there, it's 30 minutes here. So it's relatively little time exposure. And so, and again, if you go to the actual details of the protocol here, let's look at this one, for example. All right, this is double labeling. But again, antigen retrieval, 5 minutes. Rinses for a minute. Hydrogen peroxide for a minute. Triton X for 5 minutes. And again, this is Triton X for at the EM level, by the way. Again, you're exposing it just to open up the membranes just a little bit. And then you wash it off. So it's, again, Triton would totally ruin the ultrastructure of tissue. For those of you who do EM, it would totally ruin the tissue. But again, we're exposing it for 5 minutes, a relatively high concentration, 5%, admittedly. 0.5. Okay, forgot the point. There's not a point in there. Okay, 0.5. Again, a relatively low concentration.
Yes.
Okay, for 5 minutes, and then of course the primary for a little over half an hour. Again, secondary for only a little over 15 minutes. You wash here for a minute, imidazole rinse a minute. Again, in the ABC for 15 minutes. So again, relatively little exposure time. So therefore, if there's something of value in the tissue in terms of leaching out of the tissue, it has relatively little time to leach out of the tissue. I think that's probably one of the keys. So anyway, but again, you could do almost anything. These are just a few examples in the lab that we are doing. We do a lot of pre-embed and post-embed immunogold. We do single labeling, double labeling. Very, very traditionally, we do that. And so this has really worked out. Quite well for us. And again, in terms of the turnover, it takes very little time, to do it. Oh, I'm going in the opposite direction. Okay, this is a key. If you really want to process lots of samples at one time, these little processing dishes are like the best thing since sliced bread. I mean, they're really quite marvelous. So, I was going the wrong way. Sorry about that. So again, acknowledging my collaborators, the support by the Department of Veterans Affairs. And obviously Cindy has been really the leader in all of this. And just one last slide, just dedicated to Ted. Wonderful. I can't say enough. So no need to say any more about that. So I'm open to any questions that you may have or that Cindy can answer.
We have BioWave that is the version is before last version. What is the difference between this version and the last version?
We actually have the last version. What we use is the version before this. So I don't know exactly what you guys changed on there.
Oh yeah, so this is the, okay.